moment we are born, we meet our first microbes. Within seconds, we are colonized by them in a process essential to our health. Some microbes are dangerous, a few outright deadly. Tonight, follow the story of a virus outbreak, step by step. At the same time, discover the delicate dance we perform with the smallest and oldest form of life. Microbes live everywhere, outside and inside plants, animals, and people. They are single-cell creatures, such as protozoa, bacteria, or viruses. The huge diversity of microbes make all life on Earth possible. Microbes are the earliest life on Earth that we know. They are also the smallest of creatures, too small to be seen with the naked eye. Under the microscope, we see that they live in communities. They share space and food, and are continuously evolving in a finely tuned balance with their environment. And microbes have evolved together, interacting like experienced dance partners. The billions of microbes traveling around with just one person outnumber the Earth's human population. Microbial communities on the skin vary like life on the Earth's surface. Dry areas of the back are sparsely populated like a desert, while under the arm, life abounds as in a tropical forest. Microbes protect us from being colonized by harmful microbes that may pass to us from other people.
Human life without microorganisms just wouldn't happen.
We've really just begun to understand the real kinds of complexities that exist in this life that we have with these organisms that inhabit us.
Stanley Falco is a microbiologist at Stanford University who has spent his career studying the relationship between bacteria and humans.
They're the simplest free-living organisms that we're aware of.
Some live exclusively in people, some live exclusively in water, some live in the soil, some live in the sea.
Stanley Falco has a particular fascination with bacteria. Many bacteria can move by themselves, powered by whip-like flagella. Their membranes allow food in and waste out. A bacterium contains all the necessary machinery to grow and reproduce by itself. When it reaches a certain size, it divides, following the code provided by its DNA. Within minutes, the bacterium separates into two complete bacteria.
It doesn't have a brain, but it knows where it is very accurately by constantly measuring things like the temperature, the acidity of its environment.
Bacteria make up the largest number of microbes that inhabit us.
There are literally 600 or 700 species of organisms that inhabit us that we know about.
And they are part of what is called the normal flora.
Though we are sterile before birth, from that moment on, we receive friendly microbes from our mother and other people who care for us. Microbes pass to us from the hand, mouth, and breast. Our relationship with these microbes is reciprocal. We provide them a home and food, and they protect us, provide us with nutrients, and interact with our immune system.
When we leave our mother's womb, our immune system is still fairly immature and undeveloped. And really the first foreign things that make an invasion, if you will, of us are microorganisms. So the immune system begins to see and sample microbes, in a way, is learning from the microbes that it sees from day one.
Microbes colonize the skin and the mucous lining of our mouth, throat, and digestive tract. The large intestine has hundreds of species of microbes. They live cooperatively, digesting food and making vitamins for us.
There are certainly organisms that cause infection and disease, but there's a far greater number that are part of us from the day we're born until the day we die.
Some microbes, such as the ones that live in our digestive tract, are beneficial to our well-being. Other microbes, however, have been our enemies for thousands of years. Throughout history, infectious diseases have wiped out huge populations. In 1918, over 20 million people died from an influenza virus, which swept the globe. Microbial killers, like HIV, can emerge when something tips the balance in favor of a particular microbe. Other microbes, such as Ebola, the mysterious killer that surfaced in Zaire, or Legionnaire's disease, can appear when something changes in our relationship to the environment. These can be the most lethal. Around the world, few things are feared as much as outbreaks of diseases which kill swiftly and can appear anywhere, anytime. Outbreaks are not random acts. They have causes. Understanding those causes is part of the bigger story of microbes as both our friends and enemies. Dr. C.J. Peters.
We know that we're overdue for a pandemic. It may be a pandemic like 1918, where there were 20 million deaths. How do you face the probabilities that something might happen that would be devastating to humanity?
Within a week, 4 more. Dr. Stuart Levy.
One of my attendings came to tell me that he had admitted a patient that night with a disease that he hadn't seen before.
Dr. Fred Coster.
We had to rule out known viral pneumonias, known bacterial sepsis, septicemic plague.
And suddenly that patient had a cardiac arrest for no explicable reason whatsoever.
The mystery illness caused flu-like symptoms and acute shortness of breath. The victims died rapidly, within 1 or 2 days.
We couldn't at that stage predict who was going to live and who was going to die.
then seeing this kind of on a, not a daily basis, but once or twice a week.
The lab tests at the University of New Mexico Hospital searching for the cause of the disease were unsuccessful. Over 50% of the patients admitted died.
I'd come from Africa where I'd seen the devastating hemorrhagic fevers, Marburg virus, Lassa fever, and I taught them that one day we could get such a disease in this country. We don't know what this disease is. It could kill all of us.
Having all 4 members of one family come down and 2 of them die with a similar illness, of course, that's what fueled all of this anxiety.
The disease spread through the Southwest. A mother of 2 in Nevada. A man repairing his house in Arizona. A teenager in Colorado. The outbreak was serious enough to start a national alert and bring in the Centers for Disease Control.
We got the call from the folks in the Southwest, and they had reached a point where they knew they needed help. We didn't know what this was. It was totally, absolutely unknown. We knew people were dying. We knew we had a problem with some kind of disease that looked like an infectious disease. It had a very high mortality rate. We didn't have a clue what the actual organism was.
C.J. Peters is the director of the Emerging Pathogens Department at the Centers for Disease Control and Prevention in Atlanta.
We had 15 or 20 suspected patients, and it could have gone to 30, 40, 300. I mean, you just don't know when the curve starts going up until you can get a handle on things. We look for absolutely everything. We look for all the bacterial agents, the viral agents, mycoplasma, fungi, you name it.
Suzanne Jackson almost died.
They made a decision to fly me to Albuquerque. You know, I was worsening and worsening, EMT people. They were terrified. They felt that I might die on the way down. I mean, they were constantly monitoring everything. When I arrived, I was not breathing on my own.
As a last-ditch effort, doctors connected Suzanne Jackson to a heart and lung bypass unit. It saved her life. At the CDC lab, scientists were testing the victim's blood and tissue samples against all known microbial killers. The testing continued for a week without finding the cause of the disease. More people were hospitalized. All had flu-like symptoms, acute shortness of breath, and their bodies swelled up with fluid.
People would come in who could look through the window who were my friends, and they didn't know it was me, and they would, where's Suzanne? And, oh, that's her.
Within 19 days, the CDC identified this microbe, a hantavirus, as the dangerous killer, a member of a virus family that previously had only been known to infect humans in Asia and Europe. A virus is the smallest of microbes and the simplest of all living things. It causes disease accidentally, simply as a byproduct of its efforts to reproduce. Inside the cell, the virus sheds its protective coating. It must enter a living cell in order to multiply. The virus duplicates its chains of genetic material by using the host cell's chemistry. The protective coating, again, covers the duplicated genetic material. By hijacking the mechanism of the cell, the virus can make 1 million copies in hours. Once the virus was identified as a hanta, scientists had to figure out why it suddenly emerged in the United States.
We had to know where the virus was coming from. Now, other hantaviruses come from rodents. We didn't know about this hantavirus. It could have come from God only knows what.
Dr. Ben Muneta, the grandson of a Navajo medicine man, was part of the CDC investigation. He uncovered an early clue.
I went around the reservation to the traditional healers that I knew, and then I asked them about the knowledge of the mystery illness, and what they told me was quite astounding. I was listening to them describing the things almost word per word, what we were piecing together in our meetings with the CDC.
The medicine men described the deer mouse as a carrier of the mysterious illness. The Navajo both revere and fear the deer mouse. In their ancient creation story, this lightning-fast rodent spread the seeds of life throughout the world. But according to another legend, if a deer mouse enters a Navajo dwelling and sees food lying about, it gets angry at the wastefulness. As punishment, it chooses the strongest and finest young Navajos to die.
The traditional healers told me that the way that the deer mouse will take a human life is that through their urine and their droppings, and the way it passes is through your eyes, or through your mouth, or through your nose.
That's fine. I'd like to see what that means.
In Albuquerque, C.J. Peters worked closely with a multidisciplinary team of doctors and scientists, including biologist Terry Yates, director of the Museum of Southwestern Biology.
It's interesting that the 93 hantavirus outbreak actually occurred in the Four Corners region of the southwestern United States, because this is exactly the area where we have been doing long-term ecological research on small mammals, including rodents.
In the late 1970s, Terry Yates had begun preserving tissue samples of local rodent species, including the deer mouse. Now, knowing what to search for, they found evidence of the newly discovered hantavirus in even their oldest tissue samples.
The hantavirus outbreak had a very interesting effect on Native Americans nationwide, because Navajo people were very reluctant to talk about these beliefs to the outside world, because for generations people made fun of us for that. It was quite a vindication.
Scientists confirm that the virus came from the droppings of deer mice. It could pass to humans through airborne particles of dust. The CDC teams began trapping deer mice in and around the homes of the victims.
We could actually match the genetic signature of the virus in the person who had died. We could take the viral genes from his lung, and we could match those genes to the genes that the mouse in his own house was carrying.
A lot of the diseases that are most feared, by physicians at least, as being the most deadly, are those in which you have the transmission of a virus or a microbe that ordinarily lives in an animal to humans.
The deadly hantavirus doesn't make the deer mouse sick. It may even be one of its microbial friends.
We know that there must be some interaction there, or the viruses wouldn't have survived as parasites on these rodents. So there's probably an angle where these viruses are the friends of the rodents, but they're definitely our enemies, though.
There are many examples of transmission of a virus or microbe from animals to humans. Plague also comes from rodents. Anthrax from cattle. In each case, something allows the microbe to expand into a new host, Something in the ecosystem tips the balance.
Once it became clear that deer mice were the reservoir species for this new hantavirus, we hypothesized that what caused the 93 hantavirus outbreak, at least in the southwestern United States, was the El Nino-driven increase in plant primary productivity, which caused more food for mice, which caused more mice, which caused more people to come in contact with infected mice, and thus more hantavirus pulmonary syndrome.
Once the virus and its source were identified, the worst of the outbreak was contained. A vigorous public education campaign stressed avoidance of deer mice and airborne particles from their droppings. Yet, questions remain. Not least, how does Hanta kill its victims? One hypothesis is that, ironically, people die as a result of the intense response of their own immune system.
Humans seem to eliminate the virus completely, but in the process of eliminating the virus, they make themselves very sick.
When hantavirus is inhaled, it will infect cells, allowing it to reproduce. Inside the lungs, small air sacs are surrounded by blood vessels. Within the blood vessels, our white cells patrol, looking out for invaders. Hantavirus, seen here as black dots, infects the cells that line the blood vessels. The white cells sense the invader, push through, and emit strong chemicals that inactivate the virus, but also weaken the blood vessels. Plasma leaks into the air sacs. Soon, the plasma accumulates, leaving no room for oxygen.
The virus causes the cells in the body to lose their fluid components. So I was huge. My entire body puffed up to 170 pounds. I weigh 112.
The infection of human cells by the hantavirus can have a deadly outcome. But the virus doesn't gain anything if the host dies too quickly before it can pass to a new host. On the contrary, it loses its home. Other microbes have learned different, more sophisticated strategies.
Disfrute del mejor cine.
Stanford University, Stanley Falco and his students are learning how some microbes have evolved the capability to outwit the human immune system. The focus of their research is Salmonella, a group of bacteria which cause typhoid fever and diarrhea. Salmonella are different from the deadly enemy hantavirus. Seen here reproducing at 360 times normal speed, Salmonella has learned to be a friendly enemy. Unlike the hantavirus, Salmonella kills some human cells when it invades them to get food. But usually, the human host survives.
While salmonella food poisoning is common, the number of people that actually get infected by salmonella from food every year that don't show disease is much, much greater than the number of people who show disease.
Salmonella bacteria have learned to defeat the body's defensive mechanisms. They enter with contaminated food or water. In the mouth, Salmonella survive competing organisms, sticky saliva, and bacteria-killing chemicals. In the stomach, they withstand churning in an acid bath. In the small intestine, they survive bile salts, designed to rip microbes apart. They aim towards specialized cells that line the walls of the intestine as part of the immune system's defenses. Salmonella has learned through evolution how to defeat this protective cell and pass right through. Across roams the macrophage, a type of white blood cell designed to destroy invaders like viruses, or salmonella. Salmonella not only survive inside the macrophage and reproduce, they kill it and burst out to infect more cells.
This interaction, usually it's a draw. The organism got food, it made more of itself and passed on to a new person to make more of itself. And we're happy because we now recognize this organism and will be immune to it. Now, occasionally, it doesn't turn out so well for us, and the organism gets the upper hand, and it's then that we see what we call infectious disease.
One of Stan Falco's students, Denise Monak, used a microscope to videotape the interaction of salmonella with a macrophage, the killer cell.
I can't remember if this is the one that goes into a tubular...
That's right. This is one of the... Yeah. This is the mystery. No.
Right.
And when you watch this interaction, you see the bacteria coming in contact, and quickly the cell, it gobbles them up. It's almost like it's eating peanuts. I mean, it's taking them in as quickly as it can. And most bacteria that fall into the clutches of a macrophage die a horrible death. Salmonella responds by turning on new sets of genes that change the macrophage. And the end result really is the organism grows in the macrophage and eventually kills the macrophage.
Although the macrophage may die, we normally don't. Salmonella has successfully evolved to infect us without killing us, ensuring its survival. Other microbes, peacefully living inside our body, can suddenly become enemies. If we take an antibiotic to fight against that salmonella attack, our internal microbial balance can be disrupted, and some of our microbial friends will die as a side effect of the antibiotic. Then a small population of one kind of bacteria, resistant to the antibiotic, can begin to take over.
It's really found something that it must be like nirvana to a microbe, and it begins to grow. And as it begins to grow, one of its byproducts really is toxic to human cells.
The side effect of that antibiotic can be diarrhea, caused by internal microbes that are usually benign. Our dance with microbes ranges from the cooperative to the hostile. The threat from hantavirus and some other microbes is like a conflict frozen in time. It seems to appear and reappear, lying dormant in between, sometimes for centuries.
El universo.
400 years ago in the English countryside, a mysterious respiratory disease terrorized Tudor England. It was called the English sweating sickness for the high fevers it induced. There were 5 outbreaks of this dreaded killer between 1485 and 1551. The disease even changed the course of history by taking the life of young Prince Arthur Tudor. His younger brother, Henry VIII, became king, paving the way for the English Reformation. Then the sweat disappeared entirely. For over 400 years in metropolitan London, there has not been a single documented case of the sweating sickness. It was almost forgotten. That is, until 3 medical detectives suspected a connection between the sweating sickness and hantavirus.
Richard. Morning.
Dr. Vanya Gant is a physician at St. Thomas's Hospital in London who specializes in infectious disease.
These are them.
In the fall of 1997, he was approached by a medical student, Guy Thwaites, who, while researching for his graduation project, had stumbled across a historical reference to the sweat.
Which is now on this tracky side.
And he said in the late 15th and the 16th century in England, there seemed to be a disease that was characterized by fever and rapid death. And I said, go and find out about it and let's talk. Let's see whether we can build this up.
They teamed up with Mark Tavener, a 16th century historian who could translate the Old English and Latin of ancient records. The three of them set out to solve one of England's oldest medical mysteries. The sweating sickness so affected Tudor England that it was the subject of one of the first books ever printed about disease. It described all the symptoms, besides high fever and sweating.
The clinical descriptions, as best described, were of a very rapid onset. One would feel flu-like. You would have joint pains, headache, feel a little bit under the weather, and then a gathering shortness of breath to the point where some 24 to 48 hours later, you were dead.
I said, this sounds exactly like the Four Corners disease, the hantavirus pulmonary syndrome that had been described maybe 2-3 years beforehand in the southwestern corner of the United States. I mean, we knew that hantaviruses probably had been around in ancient times, way before Christ, from Chinese texts.
Guy Thwaites and Mark Tavener set out to find a broader source of information. Their riches came from grave diggers' records and old parish registers.
Parish records are important because they are some of the only surviving documents and often include a cause of death. Many of the descriptions would describe someone being well in the morning and dead by supper.
This must be it. Here it is. Yeah. Fantastic.
Then they found a description by an architectural historian of an unusually large and artful tombstone.
Yeah, they would have been there, but they've been better.
The stone marked the graves of the Duke of Suffolk and his younger brother, who both died in 1551, the year of the last recorded outbreak of the sweating sickness.
We can see the area that's depressed.
Historian Mark Tavener.
The Dukes are of particular interest because they were important enough for this to have been written about. They were studying with their tutor at Cambridge University, which is about 15 miles away as the crow flies. And as this sweat spread through the country, it struck Cambridge, and the boys, fearing for their lives, fled. And when they got here, they fell sick.
The accumulating evidence led Guy Thwaites to be more and more convinced that the sweating sickness was caused by some kind of hantavirus.
It was certainly a summer disease. It was not a disease that was associated with poor conditions of sanitation. It was scattered, it was rural, it didn't conform to any patterns of people-to-people transmission. It conformed more to some other factor involved in its transmission, and we believe that this is probably where the rodent theory comes in.
The identity of the microbe which caused the English sweating sickness may never definitely be established. However, this research adds an intriguing chapter to one of Europe's longest-running medical mysteries.
What we see in modern medicine is just a snapshot. We don't see anything other than that. We don't look at history in modern medicine. We don't get taught it in our medical schools. Perhaps we should be. Perhaps we ought to get more perspective on the evolution of infectious diseases. And we think that we are seeing them for the first time. But we're not seeing them for the first time. This is a cyclical thing. They reappear and appear, and they've done so throughout the whole of history.
For most of human history, our lack of knowledge of the microbial world left us at the mercy of infectious diseases. Microbes take advantage of any opportunity. Poverty and lack of sanitation give them those opportunities. There's no need to travel back in time to find an example. Tegucigalpa, Honduras in Central America. Perhaps the poorest country in the Americas. Many people here and in other developing countries live in conditions the industrialized world left behind many years ago. Germaine Hanquet is a physician with the international agency Doctors Without Borders. She has worked in Africa and is now working in Honduras.
In developing countries, a major problem is that the environmental, the water supply, the sanitation conditions, the level of hygiene, the level of education, the level of nutrition of the people, in general, the living conditions of the people are poor, and this is favoring the spread of those infectious diseases.
Inadequate nutrition and constant exposure to dangerous microbes can weaken the immune system. Worldwide, 50,000 people a day die of infectious diseases such as typhoid, tuberculosis, cholera, yellow fever, and malaria. Infectious diseases are the world's biggest killers.
Most of them, you can prevent them, you can treat them, you can act of them. It's not like for cancer or chronic disease for which you can't do much. But infectious disease, it's really something you can act on. And in developing countries, in Africa, in Honduras, most of the diseases are infectious. So that's really what I meet most. Children are dying.
Hola.
Hola, buenas tardes. Buenas tardes, señoras. ¿Cómo están?
Bien.
I really love this work. I feel that my job is really useful, where you are really needed, where you find a lot of disease that you can treat, that you can prevent, for which you can really do something.
At the time of Germaine Hanquet's arrival in Honduras, there was a cholera epidemic, a bacterial disease common in developing countries.
It's a very acute diarrheal disease which kills 50% of the people which are infected if nothing is done. And it's so easy to save those people. And if you treat them well, only 1% would die maximum.
The infectious diseases most problematic here are well understood. The strategies to control them have been known for years. Sadly, they still run rampant. With so many diseases flourishing here, if hantavirus surfaced, it would probably be diagnosed as pneumonia, or more likely, go unrecognized. The best defense, prevention, should be applied to all microbial threats.
I think those new diseases that are emerging or re-emerging, like hantavirus, they appear. First, you don't know who is causing them, and then you don't know which is the reservoir, and you really have to do an entire work around it to really find where is it coming from, how could we do to prevent it. You never know in advance how big the problem will be. Look at AIDS, it started by a few cases, and now it's a disease which affects the entire world.
Germaine Hanquet is a frontline warrior in the fight against infectious disease. The battle she wages every day could easily affect us, thousands of miles away.
It's clear that infectious disease, they do not have borders, they do not respect borders. Nowadays, that people are moving so much from one country to another country. Every country is at risk almost at every disease.
The largest problems are presented by infectious diseases, which are easily transmitted. As deadly as Hanta is, it kills a small number of people when compared to other infectious diseases such as cholera. Why? Because cholera can be easily transmitted through contaminated water. What would happen if the hantavirus evolved and found a way to infect people? as readily as cholera.
Lo mejor del séptimo arte.
Buenos Aires, Argentina, is a sophisticated city. It has one of the highest standards of living in Latin America. Three hours away is the peaceful community of Pergamino. Here, modern farming methods have transformed wild pampas into a fertile breadbasket. The Hanta outbreak in New Mexico had happened over 5,000 miles away, and the threat appeared to have long passed. For the local residents, it was a shock to discover Hanta appearing in their surroundings without warning in a deadly new form. Dr. Delia Enria is a physician and microbiologist expert on viral diseases. At the National Institute for Human Viral Illness in Pergamino, she noticed unusual symptoms in some patients. She didn't know it was a hantavirus. Through the internet, she received a CDC report about the virus. Since the 1993 outbreak of hantavirus in New Mexico, there had been almost 200 cases in 29 states. The CDC report triggered a memory of a patient Delia Enria had treated with similar symptoms. She paid a visit to her former patient, Oscar Michetti. At the time of his illness, Oscar Michetti had not tested positive for any known pathogen. She wanted to know how he had gotten sick.
You are a detective. You are following step by step what we call an hypothesis.
Pasaba más o menos de noche, cuando salía afuera de la
He felt that he was going to die.
The description of his symptoms, coupled with the kind of farming chores that Oscar Michetti was doing before he got sick, pointed towards a hantavirus. In September 1996, Dr. Enria's fears were confirmed. In the quiet resort town of Bariloche in southern Argentina, 4 people became ill with flu-like symptoms. They were short of breath and their lungs filled up with fluid. Tests sent to the CDC proved that the killer was another new strain of hantavirus. Over 50% of those stricken died rapidly. The outbreak caused panic. Something was happening that had not happened in the United States. People who came in contact with victims also came down with the disease. A reporter covering the story suddenly got sick and died. A doctor who had treated patients in Bariloche became sick. He traveled 1,000 miles to Buenos Aires to see a specialist. The specialist got sick. The specialist's wife got sick. All 3 died. Delia Enria suspected person-to-person transmission, something that had never occurred with any hantavirus outbreak. This would mean the virus had changed, becoming capable of jumping from human to human. If this was the case, the virus would spread easier and faster. At the hospital in Pergamino, Dr. Enria had another case.
I got a call from Dickie and Rhea at the Institute for Virology in Pergamino, and she wanted to talk about the issues around human-to-human transmission. Well, I figured that that was probably just a fantasy.
Facing skepticism about her person-to-person transmission hypothesis, Delia Enria was determined to find the source of the virus. She and her colleagues set traps in the area where the recent victim lived. Even if it had evolved to be contagious from one person to another, the starting point, the original source, still had to be a rodent. Not all rodents carry the virus, but in field work they protect themselves, as if all mice were deadly. This one had no trace of the hantavirus.
The Argentines organized a team. A couple of us went down to work with them. We began to set up a series of hypotheses about the way these people were getting infected. And we had a long list, and we went through the list, and we were able just to draw a line through each hypothesis until we were left with person-to-person transmission. Not supposed to happen with hantaviruses.
Person-to-person transmission of the virus could have meant rapid infection of nearby Buenos Aires or beyond. A neighbor, a relative, anyone could have passed the virus. Avoiding rodents would not prevent the disease.
The virus diseases that have been able to come into humans, cause problems, and be transmissible from human to human are the diseases that we know we have to watch out for.
Yet in the case of this hantavirus, the disease mysteriously vanished, at least for now, almost as quickly as it had appeared. Ironically, although the virus changed strategy, its mode of transmission, it was still reaching a dead end. Either the virus kills its human host, or the host kills the virus before it can be transmitted. Humans are not a good host for hantavirus. Yet.
You can look at that half full or half empty. Well, it only killed a few hundred people. Why are we worried about it? Or you can look at it from the point of view of it killed a few hundred people, and what's the next time going to bring? What about the genetic variability that's inherent in these viruses? Could they take off? Well, we don't know, and we'd better find out.
Research into hantavirus continues. When Hanta surfaced in 1993, it was identified fairly quickly.
Is that number 21? Right, okay.
But cases keep appearing throughout the Americas. And there's still no vaccine or cure for the disease. So far, the strain found in Argentina has been the only one capable of jumping from person to person. It is believed that hantavirus has been dangerous for thousands of years, periodically emerging or re-emerging in outbreaks. But now we have learned to recognize it.
I am completely sure this is the tip of the iceberg. Once we begin looking better for these viruses, we will find new surprise. We will find that they are causing disease that we are not able to recognize yet.
Scientists now theorize that hantavirus arrived in the Americas a long time ago. Rodents carrying some form of the virus probably migrated millions of years ago across an ancient ice bridge which once connected Asia and the Americas. Why are we just finding it now?
There are
several major things, in my opinion, that are changing our relationship with microbes and with pathogenic organisms of different kinds. Climate change and changing human land use patterns.
Now, what's happened in modern times, as shown by the hantavirus, is that human populations have gotten bigger and bigger and bigger, and we suddenly come in contact with creatures which we ordinarily didn't before.
Since the detection of hantavirus in New Mexico, 18 other previously unknown strains of hanta have been discovered in the Americas. Some fear that this is only the beginning.
We still have not encountered many of the microorganisms that are out there in nature. And the more we encroach in the environment and the more different living things that we affect, and indeed the more species that we wipe out, every time this happens, we really affect the microbial balance in the world in ways we can't understand. We are destined to live in a contaminated world. We are not going to live in a sterile environment. The evolution of our interaction between microbes and humans is in a constant state of flux. And so I think that we will, as we have in the past, constantly be surprised by new kinds of interactions that we will view as disease and the microbes will view as survival.
Understanding the relationship between humanity, microbes, and the environment is a scientific challenge for the coming decades. Using this understanding to fight microbial disease is not only a scientific problem, but a social and economic one as well. The dance with microbes has lasted as long as we've existed. While some are dangerous, most are not. We are locked in an embrace of cause and effect with the microbial world. We do something and they change. Then we change. And so it will continue for as long as there is life. Next time on Intimate Strangers.
The whole Chernobyl area is a huge petri dish.
From the